Muy bien, buenos días. Es un placer estar aquí de nuevo moderando esta sesión plenaria con Inés. Y vamos a pasar a la siguiente presentación, vamos a pasar a las células NK. So, good morning. There is no doubt that NK cells, play an important role in innate immune response to viral infections, and over the years, insights into the molecular recognition of factors targeting viral replication and also controlling diverse immune functions have put NK cells in the forefront, actually, of modern immunology. But there is always something mysterious about NK cells, and also there is no doubt that there are some gaps in our knowledge. So it's a pleasure to have today with us to Dr. Nicolas Huot. Dr. Nicolas Huot is a senior researcher at the HIV Inflammation and Persistent Unit of Institut Pasteur in Paris. And since most of the work with NK cells has been focused in peripheral blood, the work of Dr. Huot has contributed to understand the specificities of tissue functionalities of NK cells, mainly using in a very interesting way the non-human primate models. So, merci beaucoup d'avoir accepté notre invitation. C'est vraiment un plaisir. La parole est à vous.
So thanks a lot. I would like to thank the organizer for inviting me. It's always a pleasure for me to speak about NK cell and especially their complexity in the model of non-human primate infection. So NK cell it's not a mystery for you if I say that NK cell are lymphocytes. They're comprising 5 to 15% of circulating lymphocytes in adults. There are two main functions. They are able to produce many cytokines such as interferon gamma, GM-CSF, or TNF-alpha, but they are most known for the capacity to directly recognize and kill stress cells. NK cells play a role in many physiological processes, such as cancer, but also they play a role in GVHD. They are involved in pregnancy. There is some recent publication which evokes their role in their capacity to switch, to facilitate the switch to innate from adaptive immunities, and they are mainly known to participate to the control of viral replication. So there is no real marker to define NK cells, but historically the CD56 and CD16 will be used to classify NK cell into two main subsets. The first subset was the CD56 bright NK cell, which are considered as immature NK cells and are able to produce a lot of cytokine, and their counterpart, the CD56 dim NK cells, are known as the killer because they are mainly involved in the recognition and the elimination of the target cells, and they are considered as the more mature NK cells. So having said that, this, I would say, all view of NK cell have changed during the last decade, and nowadays it's well admitted that NK cells are more diverse than previously appreciated. In fact, in any individuals, we are able to count around more than 30,000 NK cell phenotype, different NK cell phenotype. And these do not take into account the fact that NK cells also are present in different tissue, and the huge heterogeneity between NK cells in any individual have pushed the field to investigate this population and to understand how heterogeneity could participate to NK cell biology. So one of the main questions currently is what is the origin of the extensive spectrum of diversity observed in human NK cell, and how are the various subsets NK cells generate in vitro. and in vivo. So there is many determinants which could participate to the NK cell diversity. Among them, there is the place where you live. In fact, the environment where you live participates to the diversity of NK cell. Pollution also participates to NK cell diversity. One of the main factors are genetic. In fact, it has been shown that according to the different CMH that you can have in any individual, it will induce diversity of NK cells. The age and the gender also participate to the diversity of NK cells. The way of life, if you are the body weight and the body mass could impact NK cell diversity and functionality. And of course, the way of life, if you are a smoker or if you do exercise, you will have a different NK cell diversity. And for the purpose of this topic, I would like to mention that, and I will focus on this aspect, that immunological history also participates to NK cell diversity. It's well admitted nowadays that, in fact, following infection, the NK cell response will generate a diversity of NK cells which could participate to the control or not of the viral infection. So what are the mechanisms underlying the generation of NK cell diversity within individuals? We could not speak about NK cell diversity without speaking about NK cell homeostasis. So the model for NK cell differentiation is summarized on this slide. It was suspected that, in fact, the CD56 bright NK cell, which correspond to the immature NK cell, could differentiate by acquire or disacquire some receptor to become a fully mature NK cells. Nevertheless, based on this model, 6 distinct development stages were identified from the NK cell precursor, which are mainly defined by the expression of CD34, CD45 array, and CD38, until the fully differentiated NK cells, which correspond to the memory NK cells, which are mainly characterized by the high expression of KIR and CD57. Of course, NK cell heterogeneity could be explained by difference between the subsets during the maturation process, but there is no unique road to become an NK cells. In fact, as you can see on this slide, from the NK cell precursor, which are here, according to different factors, NK cell could choose a fate or another one, and this choose will increase the maturation, but will decrease the capacity of NK cell to divide it. But during this maturation, they will increase their functional potential and become a fully functional NK cells. So what are the factors which drive this establishment of fully functional NK cells? There is many factors. Among them, there is genetic factor, for example, epigenetic, transcription regulation, and metabolic function are involved in this process, but also the microenvironment in which NK cells are localized. In fact, the capacity of NK cells to crosstalk with other cells, such as DC or macrophages, but and also the inflammation state will participate to the differentiation and the maturation of NK cells. So, to become a functional NK cells, you need to, in fact, the two aspects, the genetic and the environment, participate to this equilibrium, between the two aspects, participate to the fate of NK cells. So one of the main questions regarding HIV infection is how does tissue microenvironment influence the fate of NK cell and function, and what insight can we gain from understanding the localization within different tissues? So NK cell could be found in many tissues, such as the second lymphoid organ, but also in the lung, in the liver. It's not an exhaustive list, but in the bone marrow and in the decidua for women. And in fact, just to say that an overall minority of body NK cells are present in the blood. They represent only 2% of total NK cells present in individuals. So what we know about NK cell is very few amounts. And we need to investigate what does happen in deep tissue if we want to understand and if we want to manipulate NK cell in order to cure HIV. Nevertheless, investigate this. There is some recent evidence which are published 5 years ago now, which have investigated this aspect of NK cells, of tissue NK cells. And they were able to show that, in fact, in bone marrow, there is only NK cell precursor and fully mature NK cells. They were not able to find a different step of NK cell maturation, suggesting that NK cell maturation do not occur totally in bone marrow. When they look in the respiratory tract, they found only fully mature NK cells. In the lymphatic system, especially lymph node and spleen, they found mainly intermediate maturation stage of NK cells, suggesting that this tissue was the main tissue responsible for the fully maturation. The digestive tract and especially the gut also play a role in the maturation process of NK cells. And the liver is, I would say, a pellicular tissue regarding NK cell because it was supposed that NK cell differentiate totally in this tissue. Finally, to say that the blood only represents a kind of mosaic of what could we see in tissue NK cells. And the maturation of NK cells are dependent of many factors, as I said previously, which are especially their capacity to migrate among tissues, the microenvironment which is provided by the tissue, but also the presence of feeder cells, the presence of helper cells, the type of hypoxia of the tissue. If we don't have exactly the same picture if you are in the lymph node or if you are in the lung, and many other factors. So currently a model has been developed by the field, and this model could be summarized like that. So NK cells are released from the bone marrow, NK cell precursors are released from the bone marrow, they circulate into the blood to reach tissue. In each tissue, they will start to differentiate it. And having differentiated, some NK cells will stay in the tissue to become resident NK cells, and others will be released in the bloodstream. And this diversity of NK cells measured in the blood reflects, in fact, all the NK cells present in the tissue. Having said that, now the question is how a virus could impact the diversity of NK cells. Currently, it's not well known which are the specific mechanisms which influence the NK cell differentiation and NK cell heterogeneity. It may vary depending on the tissue and the nature of the pathogen. So indeed, it's not the same picture if you consider HCV infection, which targets exclusively the liver, or SARS-CoV-2 or flu infection, which targets the lung. And if you look for HIV, for example, this is the main aspect of the presentation, HIV targets all the tissue at the same time. So HIV will impact drastically the NK cell heterogeneity. Moreover, NK cell behavior could change according to the time frame of the infection. You will never have exactly the same picture if you are in acute phase of HIV infection, in which there is a lot of pro-inflammatory cytokines which are produced, or if you are in the chronic phase. Moreover, the treatment provided to fight HIV infection could have an impact on NK cells. As previously shown, according to the treatment which is provided, you can have a gain of weight, and this gain of weight could impact NK cell functionality. So now if we focus on HIV, HIV is a very complex infection regarding NK cell. In fact, as I say, NK cell HIV is able to invade many tissue, and this could impact NK cell diversity. HIV mainly replicates in second lymphoid organ, and currently the second lymphoid organ is an essential site for NK cell maturation. HIV could induce both strong and acute inflammation, which also impact NK cell. By infected helper cells, such as dendritic cells or macrophages, HIV will also disturb NK cell maturation process. And finally, HIV manipulates MHC expression, and MHC expression is supposed to be involved in NK cell education. So we think in the lab that by characterizing and comprehending the diverse fate of NK cell after HIV infection, we anticipate the ability to shape the destiny of NK cell in individuals living with HIV, ultimately influencing their immune responses. And I like this joke. The main difficulty if you want to study NK cells in human is the fact that to have a huge picture of what are NK cells, you need to get access to the tissue. And it's very difficult to get access to the tissue in a human cohort for ethical reasons and for many aspects. In this context, NHP offers a controlled environment to explore the kinetics, the phenotype, and the functional adaptation of NK cells in tissue. So in the lab, we studied 2 animal models. The African green model, which is a natural host of HIV infection. So as for the macaque model, which is a pathogenic model, I will speak about African green monkey. African green monkey have high viremia, but they do not have a chronic inflammation. They never progress to AIDS. If we compare the 2 models, natural hosts have the capacity to preserve their CD40 cell mucosal depletion, and there is no disruption of the intestinal barrier. They're also able to control the inflammation, especially during the chronic phase of infection. They preserve the structure and the function of their second lymphoid tissue, and it has been shown that, in fact, natural hosts are able to control viral replication in the lymph node, which lead to a difference of 1 to 3 log between IgM and macaque models. And finally, they harbor a lower reservoir. In the lab, we decided to investigate the capacity of HIV to invade many tissue and to look which consequence could have this impact, this infection on NK cell diversity. So to perform that, we conduct a study in which different group of monkey will have been infected with HIV. And some of monkey have been euthanized at day 4, and all the tissue were collected, and we decided to look for NK cell heterogeneity. I will not go into the detail. I will only focus on the NK cell precursor. So we perform forced GRS clustering. So forced GRS clustering, I don't know if you see it, but yes, allow, in fact, to follow each cell and the link which that there is between each cells. So based on this, we characterize the different NK cell population. So we are able to see, as you can see in red, the NK cell precursor which are in the part of this forced GRS clustering map. Then we are able to find another stage of maturation, which is here, and go on like this until the fully mature NK cell, which are characterized in monkey by NKJ2A. So we are able to characterize, in fact, the maturation progression of NK cell from the NK cell precursor until the fully mature NK cells. We go back to our tissue, and in the tissue we are able to see, in fact, that the distribution as for human of NK cell maturation stage are not the same according to the tissue. In fact, in the peripheric lymph node, as expected, we found intermediate stage of NK cell on the pellicular population, which are, for the moment, not described yet. We also, at the opposite, in blood, we found mainly fully mature NK cells. So it was anticipated that the distribution of NK cell development intermediate among tissue is not uniform, as for human, in NHP. But what role, if any, could HIV play in influencing the even distribution of NK cell among tissue? This could encompass, in fact, change in their function, distribution, and overall effectiveness due to the presence or influence of HIV within the immune system. So, just I would like to focus on one population, which are the NK cell precursor. They are defined by CDC34 positive, CD7 positive cells. And you can see that the dynamic of this population in the different tissue change with the time. In fact, as you can see, we found an increase of this population during at day 9 following infection in the bone marrow. There is no change in the blood, but there is an increase of this population also at day 9 in the blood, suggesting that there is a release from the bone marrow in the circulation following infection. We are surprised to see that this population disappears in the lymph node. And we don't find any change in the both spleen and liver. So having characterized the dynamic of this population, we decided to look for their phenotype. So based on also, we perform multidimensional analysis, and we are able to define 80 clusters of CD34 positive CD7 NK cell precursors. And we are able to show that, in fact, the dynamic and the phenotype of the cells change during the time course, and I will focus in the lymph node for this slide. You can see easily that, in fact, at D0, the main population found are in green, and this population start to disappear to see the purple population. Then at D7, we found a brown population which start to increase. At D9, the brown population migrates to become multiple, I would say, color population with different phenotype. And at D14, there is a kind of resolution, and all the NK cell precursor become at the base, the phenotype of the NK cell precursor go back to the baseline level. So this study is still ongoing, but we expected that the fluctuation of CD34 positive, CD7 positive NK cell frequency potentially intricately shapes the differentiation and functional property of NK cell in non-human primates. Just a few words to say that we are able to transpose what we observe in macaque or in AGM to human. We are surprised to see, in fact, which wasn't expected, that NKG2A was rapidly expressed on NK cell precursor, especially in the macaque model, as you can see when compared. Whereas in the natural host, NKG2A do not appear to be increased on NK cell precursor. We decided to look in human if we could see exactly the same phenomenon. So no, sorry. Sorry. So now I will focus only on the natural host, on the lymph node. So as I mentioned previously, in the lymph node there is a huge control of viral replication, and this is mainly linked to the fact that, in fact, in African green monkeys there is no viral replication in the B cell follicles. Whereas in macaque, as you can see by the red dot, a lot of viral replication occurs in the TFH cells. We are able to demonstrate that this absence of viral replication in the second lymphoid tissue, especially in the B cell follicle, was due to the accumulation of NK cell in natural host, and this accumulation was dependent of the CXR5 expression at the surface of the NK cells. So to confirm the capacity of NK cell to participate to the control of replication, we decided to deplete NK cell from the natural host by injected anti-IL-15 antibodies. The injection of anti-IL-15 antibodies allow, in fact, the good depletion of NK cell both in blood and second lymphoid tissue. And this depletion leads to an increase of viremia in African green monkey and an increase of the cell-associated viral RNA in the second lymphoid tissue, confirming the role of NK cell in the control of viral replication. We next decided to investigate how NK cell acquire the capacity to recognize and to kill the target cells, and to perform this, we decided we developed a model in which, in fact, NK cell maturation could be defined in a four-step event. So from the less mature towards the more mature NK cells. We confirm this model in different species of non-human primate models, such as cynomolgus macaque, rhesus, baboon, and African green monkey. And we use this model to investigate, in fact, the terminal differentiation of NK cells. And as you can see on the pie chart, following infection, there is a huge change in the NK cell phenotype during the time course of infection. And in fact, in African green monkey, we are able to demonstrate that this change was linked to an increase of NK cell maturation at day 9, and whereas this maturation do not occur in pathogenic models. We are also able to link this maturation to the capacity and to the generation of adaptive NK cells, which are able to participate to the control of viral replication. So now I will focus on the image chain. So having characterized the capacity of NK cell to migrate into the B cell follicles and to recognize the target cell, we decided to investigate how NK cell could recognize and eliminate the infected cells. And we focus our attention on the MHE class molecules. So non-classical MHE class 1E is few expressed at the surface of the cell, but its upregulation could occur in various contexts. Indeed, cellular stress or inflammation, such as oxidative stress, heat shock, or inflammation, upregulate MHE expression at the surface of the cells. Tumor cells are known to express a high level of MHE. It has been shown that viral infections such as HIV could upregulate MHE at the surface of the cells. And finally, cytokine, especially interferon gamma, increase MHE expression at the surface of the cells. So the two main ligands known to recognize MHE at the stress cell are expressed by NK cell are NKG2A, which deliver an inhibitory signal to the cell, and NKG2A is in human present on 20 to 90% of NK cells. And their counterpart, the NKG2E, which deliver a positive signal allowing the killing of the cells. And NKG2C is represent only 5 to 50% of NK cell in healthy individuals. Interestingly, those two markers are mutually exclusive. So we speculate that MHE engagement with NK cell through this receptor contribute to the regulation of NK cell function, especially during HIV infections. So just a few models so when a cell expresses classical MHE class 1, which are represented in blue on this picture, after a stress signal, the cells will downregulate MHE class 1, and this downregulation will occur concomitantly to the upregulation of stress ligand for NK cells, such as MIC-A or MIC-B. Two events could occur at this stage. There is no upregulation of MHE. In this context, NK cell will be able to recognize the stress cell and kill it. At the opposite, if the stress cells start to upregulate MHE, the NK cells will, two cases could occur. If NK cell express NKG2A, there will no event of killing, and if NK cell express NKG2C, it will link to an event on killing. So, based on this model, MHE is often referred to as a backup molecule, which allow, in fact, the resolution of a stress event in a cells. I will move. Sorry. So we were able to demonstrate, in fact, that in African green monkey, MHE, TFH expressing MHE are downregulated and controlled during the viral infection, as you can see on the left part of the slide. Following infection, there is a downregulation of positive TFH expressing MHE, whereas the level of this population was very high in macaque models, suggesting that in fact NK cells are not able to kill the infected TFH cells. So we perform in silico and in vitro analysis to show in fact that the peptide present in the signal peptide on the ENF protein could be loaded and stabilize MHE expression. So we developed this model in which infected CD40 cells upregulate MHE at their surface, and by upregulating they load peptide. And we perform a cytotoxic assay in which NK cells will be put together with target cells which are loaded or not with a peptide. And you can see that, in fact, when cells are loaded with peptides, NK cells are not able to kill the target cells. And this mainly due to the fact that during HIV infection, more than 90% of NK cells expressed by NK cells express NKG2A, which deliver an inhibitory signal. We perform the same experiments using non-AGM NK cells, and we don't find, in fact, we are surprised to see that AGM NK cells are not able to kill, despite the fact that, as for in MAC, they express a high level of NKG2A. So we suspect that, in fact, in AGM, NK cells have found a way to counteract the inhibitory signal. And to perform that, we perform a deep sequencing analysis, and we found one candidate, which is called TEMIS. So I will not go into the detail, but TEMIS are found highly expressed in NK cell in AGM. So just a few words on this. TEMIS have been previously shown to participate, in fact, to the TCR avidity in T cells by playing a role on sequestering HCP1, which is an adaptor molecule facilitating the transduction of the signal when the TCR is engaged. And we decided to see if, in fact, TEMIS could also participate to the inhibition, to the regulation of NKG2A as for the TCR. So, as you can see here, when we put NK cell without any target, TEMIS is not expressed, and when we start to put a target, we found an upregulation of TEMIS. And this is also true if we put NK cells, target cell with peptides. So we speculate, in fact, that TEMIS is upregulated in AGM and facilitates to participate to the blockage of the inhibitory signal provided by NKG2A, facilitating the killing of the target cells. So this work is still ongoing. But we have performed some experiments in a model of SARS-CoV-2 persistence. And we can see that, in fact, in the monkey where the virus persists, NK cells are not able to produce TEMIS, suggesting that the same mechanism is involved in SARS-CoV-2 persistence. So, just a few words on NK cell reprogramming. So, NK cell reprogramming is a goal, in fact, to achieve, according to me, to achieve HIV eradication. There is many ways to induce NK cell reprogramming. Among the main studies are the cytokine stimulation. So many interleukins, such as L2, L12, 15, 18, or 21, have been found to boost NK cell cytotoxicity and proliferation both in vitro and in vivo. In this context, in collaboration with Mirko Paardini, we have performed a study in which monkeys were treated with L21. And we are able to show that the treatment by L21 increased NK cell maturation, also increased NK cell cytotoxic activity, and increased their capacity to recognize and to kill target cells. At the end of the L21 treatment, we are able to show that the viral reservoir was lowest in animal treated by L21 than animal not treated, but we do not see any difference, or a small difference regarding the rebound after ATA after ATA. So another way another approach to manipulate NK cells, in fact, are to block the inhibitory receptor, such as NKG2A. There is an antibody which are currently available, which is called monalizumab. We can also boost the activating receptor using BNAbs, for example, to increase this IDCC activity or to use TLR agonist. Finally, we can also genetically modify NK cell to generate CAR NK cell to target the infected cells. But there is remaining challenge, such as the induction of memory NK cells following vaccination, or to target regulatory NK cells to improve humoral response. Nevertheless, we strongly believe that we probably need a combination of strategy to increase the capacity of NK cell to eliminate the infected cells. So just a few words, CAR NK cells are engineered to target HIV-infected cells and to enhance the immune response. There is many ways to generate CAR NK cells. You can use NK92 cell lines, which have been shown to be safe for individuals, but you can also isolate fully mature NK cells from PBMC and to manipulate them genetically to express a CAR receptor. You can also differentiate NK cells from the precursor. In any case, there is many caveats regarding CAR NK cells. The main problem is we need to identify suitable targets. We also need to be sure that the CAR NK cell will persist and survive in the host. We need to avoid any off-target effects. You should have in mind that, as I previously shown for macaque, HIV highly impact NK cells, so this impact of NK cells suggests that probably this strategy will not be sufficient to generate fully functional CAR NK cells. We need to think also about tissue accessibility. Indeed, if you inject CAR NK cell, you must be sure that the CAR NK cell will be able to invade all the tissue. And finally, we need to be sure that CAR NK cell could be stay and persist in the tissue to fight the infection. So in conclusion, an animal primate model of individual insight into reprogramming NK cell against HIV, the use of NHP has significantly advanced our understanding in NK cell biology. While NSP research have unveiled encouraging prospects, further investigation are essential to translate this finding into effective application. And they continue to utilize with pivotal in advancing our quest for innovative, effective, and target therapies. I would like to thank you for your attention, all the members of the unit, all the persons which are involved in the NHP study, and I will be happy to take any questions or comments.
Nicolas. Tenemos tiempo para un par de preguntas. Pepe.
Hi. Bonjour, Nicolas. Pepe Alcami from Madrid. Very nice talk. I have two questions. One is more pathogenic. Are natural killer cells depleted or their function is decreases during acute infection? In humans, is that been described? And that could be a mechanism of lack of control if treatment is started later.
Alors, regarding the depletion of NK cell, effectively there is there is a strong depletion of NK cell in the blood during the acute phase of infection. It's not clear if this depletion is linked to the fact that NK cells migrate into tissue, especially in the gut, or if they are killed. And now we also know that in fact during the acute phase there is also a decrease in NK cell capacity to kill. Until day 28, and this phenomenon, the capacity of NK cell to kill is not restored for all the monkey. I don't know for human, but for all the monkey, it depends on the monkey.
Okay, and because that could be a role in post-treatment controllers, not the control by NK, if the function is preserved because people are treated in early infection, that could be one mechanism to explain the post-treatment control. Yeah. I understand. Yeah, the second question is more technical. You mentioned that there is no co-expression of NKG2A and NKG2C in primates. In humans, we can see this co-expression, but as far as I know, there is not a good antibody against NKG2C for human non-human primates. Is that
normally real? In human, it's very debated. In healthy individual, I mean, if you exclude CMV is very tricky with that. But in healthy individual, NKG2C and NKG2A are segregated. Normally, they shouldn't be on the same cells. Effectively, in HIV individual people, we also found some cells co-expressing both NKG2C and NKG2A. It also occurs in some CMV positive people. For the moment, we don't know exactly why. We don't know if it's due to the fact that NK cells are blocked in the maturation stage, and they are not able to resolve, but okay. And effectively, you're right, in NHP we have some difficulty to investigate NKG2C, mainly due to the fact that there is no antibody available. But we perform a lot of single cell study, RNA single cell study, or deep sequencing of different subsets. And we are surprised to see that in fact NKG2E is very, very low expressed in primates when compared to human.
Okay, thank you. Thank you for the talk.
¿Alguna pregunta más? Nicolas, just a general question. Just NK cells were named almost 50 years ago. At that time, everything looked pretty simple. They could kill infected cells without any kind of activation. Do you think that this concept still holds, or it's we can consider not that natural killer cells?
I think that NKSL recognize a stress event, not directly the infection, but very stress event. And this is the equilibrium between the activatory receptor and the inhibitory receptor, which leads to the killing of the target cells. I think that the capacity of NK cell to kill a target cell is depending not on NK cell, but on the target, are dependent of the target cells. Meaning that if a target cell are not in a good feature, they will be killed by NK cells.
Okay, and also, is there any development in small molecules, or I mean, might be with biologicals, just to, I mean, to increase this targeting of NK cells towards the viral reservoir or viral load? Are there any molecules, small molecules that are just
The problem with the viral reservoir in HIV is that there is no specific marker for this reservoir. So it's difficult to imagine a molecule which facilitates the link between the non-productive reservoir and the NK cells. I know that there is some study in concert or in the concert field where they found, in fact, some molecules which are expressed only by concert cells, and they develop many aspects of such, for example, aptamer, and they are able to facilitate the linkage between the NK cells and the target cells. But in HIV, it's more complicated.
Okay, thank you very much. Bueno, pues muchas gracias Inés, ha sido un placer. Muchas gracias a los speakers, excelentes charlas, y muchas gracias a la audiencia por vuestra participación. Cerramos la sesión.